again. Sifting 925. That one made my day. What a way to make a living. Woo! I am going to be sifting again today, but I'm figuring you guys are all bored as heck with watching my sifting videos. So, here's what I'm going to have you do. I'm going to start my new investigation 2. Perfect. You can see this all right. And it's going to entail some cereal rye. I think it's called cereal rye. That's my measure cup. I've got 4 trays. 1. 2 is going to be to start. And then we're going to put down some newspapers. I'm just going to show you what this investigation is going to look like. I need a small tub. That's okay. We won't worry about it right now. The only thing is that I'm going to do, because this is the first part of the experiment, I need to get rid of some of the cereal right here. I'm going to just measure out. You know what I need to do? Well, you guys can use other different measurements. I'm just going to use this little cup right here so that we know both the same amounts of seed are used in the experiment. So, I at first did this experiment and I thought, oh, I'm probably not going to see any difference. There's probably going to have to be some sort of scientific tests done to see a difference. And so I did it originally and I was kind of like throwing it together and wasn't being crazy specific. And we still don't need to be crazy specific. But what we have now is basically two small amounts of the rye. I'm going to put the extra on the plate. And then in jars. Ooh, I lost a couple seeds. Oh no. No, the experiment is still going to be fine. We don't need to, technically, since we're not going to be writing some sort of a journal article or paper, we're just citizen science here for the people. And we want to know. So, 2, same seed. And then 1 of these I'm going to fill up to, let's go with 2 cups. This is actually rainwater, but I've collected it and put it in this tank. So let's go with 2 cups of rainwater. All right. Now, here's the problem. I'm not going to be able to use 2 cups of rainwater in this one. But what I'm going to do is I'll fill this little cup with castings. Now, these are sifted castings. These castings haven't been sprayed with mosquito bites or anything. They're active. They are a little bit dried down because of the the sifting process I wanted to. Now, that's a lot of castings in my opinion here. But this is going to have all the... I was looking for my coffee. I thought I had a coffee. Well, anyway, I did have... everything's a mess here. So this castings is going to go into this jar only. Okay? So then we'll add the approximately... We'll just bring it up to the 1 cup. Okay. The 1 cup. I'll see where that fell on there. That would inoculate this tray. So we don't want to inoculate the tray. But not everything's sterile anyway. You know. And so that's going to get in those seeds. That's going to have the fungus in there, the protozoans, and the microbes that we want to go ahead and incorporate in with those seeds. So from the time they germinate, they're going to have everything there that they need in order to bring them the nutrients from the soil. And that's why we want our to make our what are called biocomplete seeds. I'm just looking for something to stir with here real quick. I just want to stir it up. Okay, and then we're only going to allow them to be in there, say, about 12 hours. So we don't want any anaerobic conditions to start. But the microbes are already alive and active here because nothing has went dormant. And in that amount of time, these seeds are going to soak up the water. They're going to, maybe there's rhizophagy going on in the seeds. I don't know. I'm not, oh my goodness, I am not like mad scientist on that, all that plant stuff yet. I'm getting there. All right, so that's it. That's the experiment for part 1. And then these will set overnight, 12 hours basically. And then what I'm going to do, tray 1, tray 2, is we'll filter out the water from both of these. What we'll actually do is go ahead and soak both of these with water, rainwater, because we want those moist for the seeds to germinate. And we don't want to continue to add, so we're just going to do the seed inoculant portion right now for your experiment. So we're not going to, so both of the newspapers basically have ink. We can say that the ink helped one grow better than the other, things like that. Now you might get me when we do our covers because my 2 covers are different colors. I'll see if I can find 2 of the same colored covers. I can. They're right over here. I'll grab them and I'll show you what we're doing. And you don't have to do it exactly like I'm doing, but 2 separate trays need to be used so that we don't have cross-contamination here. When we do, I'm just going to go ahead and I'll filter it through something, catch the seeds, and I'm going to just lay the seeds out on top of the newspaper, and then this one I'll filter. I can pour those, this rain, I'm going to stir this other one with a different, they really don't need anything to stir them. They can just stir around in that rainwater and soak up that water, okay? That's the only difference between these 2. Then we have our vermicastings in here. So all that microbial life that we have in the vermicastings are now going to incorporate with those seeds. And when we put them on our paper, we'll see what they're able to do. Now, there is obviously nitrogen, phosphorus, and other things in that water. But that's just another reason why we can promote our vermicastings, because there are other now we could just get plain soil or dead soil or whatever. So we're getting these nice and soaked up, and then tomorrow, whatever's not soaked up before we add the seeds, we can dump any extra water off. Here's another thing I'm going to do just to prepare a little more. We're going to put one on the top of the seeds as well. So it's basically like putting them in a soil. So they're going to have soil on the bottom and soil on the top. Only it's just substrate newspaper. Okay, so we'll let those 2 soak while the seeds are soaking. All we're going to do tomorrow is take the covers off, and we're just going to sprinkle the seed in there. And then we're going to put the cover back on. And then on our other one here, we're going to just sprinkle the seeds on there. Okay. So we're just soaking these newspapers real good so that the seeds will have moisture. There's no way there's going to be any cross-contamination because we're in separate trays. There we go. Now everything's going to be good and wet. And then what we'll do in the seeds, we'll let them germinate. And we'll check them, say, every 24 hours. So we'll see who's germinating first and who's, how many more are germinating. One versus the other. These are some of the things we're going to look for in our investigation. Is there more germination in one compared to the other? The other thing is, I've already done this experiment once. I noticed in one of them, there was a lot more mold growth. So understand that when you put your seeds in the ground, mold can start to decompose that seed, especially when there's a lot of moisture in there. So we don't necessarily want that. So preventing the mold may be a benefit. So let's watch for the mold. And then there will also be other types of funguses starting to grow. And in my last experiment, I have theories as to why I noticed some seed so seed grew better than others in the trays. So stay tuned. I can link in possibly in the video. I'm not sure how I'll do this. I think I'm going to just go ahead and keep you guys posted on the experiment, and then as it's going, real, just upload a quick video. Instead of waiting for the experiment to get done. I've already done one, so I know what's going to happen, but I think if all you guys want to do one too, because then you can actually have a visual for your own clients, I suppose. More telemarketers. To show them the benefits of your vermicastings. Okay, the other experiment or investigation y'all can do is when you're adding, so I love the sheep wool experiment. Big thumbs up to DNA worm castings for going and doing that and giving it a try. You will notice those castings, I think from my past investigation, the best place for that wool is in the nursery. It retains moisture fantastically, and those castings, if there was one thing I had to pick to describe it as when you get that full bin of castings, it feels like if any of you have ever had a foam rubber pillow, those castings are like a rubbery type. I don't know. I don't know what you would call it. It's just wild. So they're super cool. And in the older bins, which are more mature, it'll break down faster. But yes, you'll see those fibers just start to come right apart. So thanks a lot for trying that investigation out. We'll be doing a lot more because why? We the people want to know. And thanks for the shout out there in Australia. That's amazing. So check him out, guys. He's trying hard too to do some real world science investigation and bring the truth to you guys here in the citizen science community. I think that's it for this short video. We're gonna finish it up tomorrow morning, and then I'll just do some every 24-hour research on how it's going. So it may be a month before the whole process is done, but there's another investigation y'all can get started. Oh, the other one is when you start doing these experiments, testing your castings as well to see what the nitrogen levels are in your castings. So what these farmers are going to be needing is either a fungal dominant or bacterial dominant substrate to apply, depending on which cash crop they've decided to grow. Excuse me. So right now, like at different bins, you can start to manipulate what substrates you're adding, what your food sources is you're adding, and then checking to see whether your substrate is a fungal dominant or a bacterial dominant, because when they're going to be looking to buy your castings, they are going to probably request whether you have a fungal dominant or a bacterial dominant casting. And then they will purchase that and extract that biology off and apply it to their field. So these are the kind of research investigations that we need to be doing. So we know how to produce that product for them in the future, possibly if we're forced to only use natural fertilizers instead of synthetics, even though we want to do that. And we want to try to avoid the nitrates. So everything's kind of going towards that now. But that's some more investigation we're going to do in the future. But I'm just giving you a heads up as to what I'm seeing and what I'm hearing. And because I'm doing the first real life out in the field investigation myself with my own eco-friendly fertilizers, converting the conventional farm into a microbial farm where we are going to be sequestering that carbon. 1, 2, we're going to try to produce our nitrogen real time in pockets in the soil where the nitrogen fixing bacteria can be harbored and convert that nitrogen from atmospheric gaseous nitrogen into NH4, which then they can take to the plant. So it's going to be really, really exciting to see if that works. Oh, we're going to go to the no-till method. You got to do the no-till method so you don't tear up those pockets where those bacteria are fixing that nitrogen. Anything else I can recap you with? I'm going to be getting behind weekend, getting prepared for lambing season. That is going to be fun. Those cute little baby lambs. So, that's what you need to be thinking about in your casting. So when you start adding certain sources like your sheep wool, hair, I'll alpaca fibers, those are high nitrogen sources. So when you guys sell those castings, you may want to have that tested so you can show people, look, you're not just getting carbon here, you're getting nitrogen. Because, you know, if you're just feeding them the carbon all the time, the cardboard or whatever, you might not have as good a nitrogen ratio in there. And so it may be that you could bump up your price a little if you can prove to them that you've got really high nitrogen castings. Okay, I just put a bucket of my, I just called it vermicompost. It was probably made new castings into the thermophilic pile. And oh my gosh, the Soil Food Web Mentor was like, oh, we are all having such a hard time finding, well, he said a high nitrogen cow poop. Well, and I know why my cow poop is so high nitrogen, which all has come back to the Soil Food Web, because I'm trying to produce a very good quality product for my animals to eat. And so they're eating an alfalfa clover, very high protein, high nitrogen food source. So when it comes out the back end, my nitrogen's super high. I also pick it up super fresh, so it's not already decomposing and losing that nitrogen. And also, I think maybe the reason that thermophilic pile, oh my gosh, we turned it and boom, by the next morning, it was up to almost 160. That was less than 12 hours. I did wrap it though, so I retained what moisture that I had, because it was just, not moisture, I'm sorry, heat. It was at 170 and we had to turn now because it was getting too hot. But I thought, oh gosh, if we turn, we're going to lose heat. So I wrapped it that night because we're still getting pretty cold at night. So I retained that heat and it, boom, it got right. It went down to 100 degrees, boom, right back up to 160 again. So I took the wrap off in the morning. And since it's only a 60 bucket pile, our ambient temperature dropped down. So we're doing good now. But that thermophilic composting with the right ratios is, and the right moisture is powerful. That's all we got to say, powerful. So I think it might have been the vermicastings adding that extra boost of high nitrogen, because it was out of the bin where I did have a ton of fleece in the bin, which got broken down. So who knows how much nitrogen that fleece is providing. I still need to do the research on that, but I will try to find some companies to have that sent to so they can give me the NPK reading on it, and then we'll know the difference, because we can probably bump that up depending on what substrates we're adding in there. So think about that and come up with some investigations you can all do. Content. We need content. I think every video I'm putting up now gives me one AI subscribe. I just think they're AI. That's all I can say. Thank you so much for watching, you guys. Let me know you hung out and with me today. And stay safe. We hope to see you in the future.